Ladies and gentlemen, hello. Welcome to the Renaissance podcast. I am Christopher Martin Burns, joined this week by my guest, Mr. John Allen, General Partner of Coddle Creek Capital down in Charlotte, North Carolina, or Kannapolis, North Carolina, I should say. How are you, John?
I'm doing well, Chris. Thank you. I'm delighted to be on the podcast.
Delighted to have you here, my friend. I been working with you for a little while, not too long, but I came and visited your facilities down in North Carolina a couple of weeks ago, and was blown away by what I saw. Was incredibly inspired by the work you're doing, the fund is doing, your portfolio companies are doing, and was inspired by your story. Your drives and where your focuses are, and how, I guess, different you are to most life sciences funds in my mind, because the amount of added value that you can bring is quite extraordinary. So I wanted to have you on here, and kind of put your story out to a wider audience. I think it's a very interesting one. I think it deserves some light shone on it. So, very pleased to have you here. And would like to maybe start with just winding it back to the beginning and talking about just your story personally, where you grew up, how you got to where you are now, and then we can dive into talking about Coddle Creek.
Great. Well, I'm a Southern boy in U.S. terms. So I grew up in the South, and I grew up in a small Southern state called South Carolina,
Mm-hmm.
uh, in Union, South Carolina. And my father was part of the cotton industry, mill industry in South Carolina.
Mm-hmm.
And so, um, I like to say I grew up on the cotton trail. Um, and so my father had done a variety of jobs with a large textile manufacturer in South Carolina called Milliken. And I had the privilege of really growing up in a state that um, had a number of challenges, but had a number of opportunities relative to kind of its underlying manufacturing base and so forth. So we saw with NAFTA frankly the destruction of a lot of manufacturing base, and so that
Yeah.
included the textile industry. So that really impacted South Carolina. It also impacted Kannapolis, North Carolina, the current city that we're in. We actually had the largest layoff in North Carolina history in Kannapolis. It was the old Fieldcrest Cannon Mills, which is a world famous mill for some of the beautiful towels that they used to make, as well as other fabrics and so forth. And growing up in South Carolina, I had the privilege of going to Wofford College, which is a small liberal arts college in Spartanburg, South Carolina, which is actually the headquarters of Milliken. And I majored in chemistry. And the name of my chemistry building was called the Milliken Science Building. So it was funded by Mr. Roger Milliken, the owner of the mill,
Right.
or mills. And so I grew up in South Carolina, went to Wofford, majored in chemistry, and then after college, went to work with a pharmaceutical company called the Upjohn Company. And so I did a variety of jobs inside of Upjohn, sales marketing across a broad catalog of women's health care, oncolytics, primary care drugs, etc.
Mm-hmm.
And I had a biochemistry professor, Dr. Scott Mara, who was evangelizing me back in the late 80s and early 90s. Hey, you've got to go into biotechnology. And at the time, I didn't know what biotechnology was, but I was just dumb enough to try to follow his advice. So he wanted me to go to work for these small companies like Genentech with 48 employees and Amgen with 24 employees. And
Yeah.
so I started putting fellows out there trying to make a break into biotechnology. And in the mid 90s, finally got a break with a small firm out of San Diego. Now, Chris, you know San Diego is a biotech hub. When
Certainly
I was
is.
in San Diego, it was not a biotech hub. Um, As a matter of fact, the company that I was with, Agron Pharmaceuticals, was the first profitable biotech company in San Diego County.
Wow.
Um, yes, and so
So
you know La
you
Jolla.
basically started it all.
Well, I didn't start it. I went to work with five professors out of the University of California that had really developed some interesting technology around rational drug design, computational chemistry.
Hmm.
And so today we do a lot of drug discovery on the computer. We call it in silico development. And we were the first drug that was ever developed on the computer that was approved by the FDA. And we had targeted at that time the HIV. So you're too young to remember when HIV was a death sentence. But for those in our audience who remember Magic Johnson and Greg Louganis
Hmm.
and how devastating HIV was, because it was a death sentence. Everybody died usually 7 to 8 years after contracting it,
Yeah.
and standard of care was AZT D4T or AZT 3TC, and then we were part of the triple cocktail revolution. So our company developed a product called Nelfenavir. The brand name was Virusept, and I was the first person hired in the field-based commercial operations to help build a sales team to launch that drug. And we wound up getting that drug approved, and then wound up having the largest biotech launch ever at the time. So we did 335 million first year. That's since been eclipsed by a number of biotech companies, but at the time, for a small biotech company that decided to compete against Roche, Abbott, and Merck, which also had protease inhibitors, and for us to be fourth to market and win 59% market share first year was a big accomplishment for our team.
I'll say. I will say you are a hero for doing that, John. I think I
Well,
speak on behalf of everyone in the world for saying that.
I was delighted to be a participant in a revolution that Peter Johnson, and who was the chairman and CEO of the company, and others had put together. So, I learned a lot through that experience. And when you look back, you make some pretty good money, particularly on the options and that type of thing. But the memories you have are really the lives that you changed.
Of course.
And one of the interesting things that we did at Agron is we had a preceptorship at San Francisco General Hospital, and we had our sales reps go through that so they could interact with patients. And by the time we had launched our drug, a number of the patients that we had precepted with had passed away, and a number of the patients were able to go on our therapies, and then wound up having stable disease afterwards. So it was a real blessing to see patients extend their life. And first month we put 89,000 patients on our drug, so it was a big deal, and lots of good memories, lots of great relationships from those times at Agron.
Yeah, that's incredible. It's incredible. I'm curious about the approval process that you went through for that. Like, what was the? I presume the appetite with the FDA must have been like, let's get this through as quickly as possible at the time. Um, or did you still come up against a lot of the same hurdles that many do now when they're trying to get stuff approved?
Well, one of the distinctions of Nelfenavir, and I believe that this is the case even to today, that from what we call bench to bed. Okay, so from the first time the molecule was physically made,
Mm,
right? So when a molecule is made on a computer, it doesn't mean that we can physically make it, right? Um, so we've got to make the molecule to the time that it's at bedside or approved by the FDA. To date, is still the fastest approval ever from the FDA. It was 3 years from the time that it was synthesized to the time it was approved. And so I was real proud to be part of a team that had success both in a quick FDA approval and
mm.
then had a spectacular approval and adoption in the marketplace.
It's extraordinary. Absolutely extraordinary. Um, so from there, did you move on kind of after it was in the market, or did you stay in San Diego? How I mean, how what happened between then and you getting back to the Carolinas?
Yeah, so I commuted to San Diego. I managed the southeast region to start with on the sales and marketing side, and then was promoted after in my fourth year was promoted to head our medical science liaisons that were responsible for phase 3B and phase 4 research, which is where I really got introduced to more the clinical development side of the business. And so I had the privilege of working with some really smart liaisons that were helping us develop commercial studies. And our group interacted with Harvard, Hopkins, Stanford, all the major, tier one research institutions. And we prosecuted research that we felt like were answering important commercial questions. So, and then after that, we were bought. I was there for 6 years, and we were bought by Warner Lambert for $2.3 billion, and then 9 months later, we were bought by Pfizer. So it wound up being a Pfizer asset, still a Pfizer asset today.
Yeah, and one that has appreciated in value, I would hasten to add. So it continues to be a success story. Okay, so let's move on to Coddle Creek Capital. And again, let's start at the beginning. How did you start the fund? Where did the idea come from? Talk us through that and how it fits really in quite an extraordinary way with the research campus that you're based on and the local area in general.
Yeah. So after we were bought by Pfizer, one of my dear friends and a guy I went to college with was a broker and with Smith Barney, and the consulting group services, which primarily worked with high net worth families, and he had a vision that with the advent of technology and the internet, that advisors were going to want to go independent. And he knew I had an interest in finance, so he asked me to come start a finance company of all things. Now, I was asked to start a finance company with him, not because I had prowess in finance, but I just happened to be his richest friend at that time. And so he was like, okay, buddy, let's go. You got some money. You want to learn finance. I'll teach you finance, and you can help seed our initiative here. So he and I started a finance
some of that
company.
exit money over here, John.
Yeah. So we did that. I started with my dear friend Greg Leakley, and we two of us started with a business plan. We grew the company to 112 people. We started obviously with no assets under management, got to 24 billion in AUM, and we were building an independent family office platform. So today, the kind of the leader in that space is a company called Adipar, which is out of Palo Alto. But if you look at Adipar, we were 10 years earlier than Adipar. And so we developed a lot of tools, which included evaluating asset managers. So we had 330 basically Coddle Creek Capitals on our platform, right? So we had independent asset managers that we made available to these family offices, and we had a staff of 6 CFAs that would do due diligence on the various asset managers, making certain that they were holding to their investment thesis and that their returns were commensurate with the risk that they were taking, and that they were creating alpha. So that's really where I learned the craft of finance and had learned really asset management. So I did that for 8 years, and then after we sold that company, I decided to move back into really what my passion is, and that's working with principal investigators, early stage, novel mechanism of actions, very interesting science, and then bridging what is called in the investment space the valley of death. And the valley of death is the gap between a new scientific discovery and commercialization. And that gap is where a lot of assets die. And that's frankly, that's where we play. We feel like we understand novel mechanisms of actions. We think we understand how those can impact human health. And then, unlike principal investigators who've not prosecuted IND campaigns, preclinical campaigns, and commercial campaigns, we really come alongside them and bring a team in to help them commercialize these novel concepts. So Coddle Creek is really interested in early stage assets. We're typically first investor in with a number of these assets. And we play active roles on these boards. So we're not passive investors. We're not following larger investors. Not that we wouldn't co-invest with some of the leaders like an Arch or 5AM or an Atlas or Hatteras, those types. But we typically like to be on the ground floor. We feel like we understand novel technology really well, and we're specifically embedded on the North Carolina research campus because it's really the hub of the areas that we're interested in. So Coddle Creek is primarily interested in 6 sectors. We're interested in genomics, nutrition, neurology, oncology, microbiome are kind of the sectors that we really focus on. That's kind of code word for longevity. Now, there's a lot of snake oil and a lot of promises in longevity, but there's also a lot of science that's coming along in the area of longevity, and we think that these initiatives to help extend human life and make the quality of life, particularly later in life, better, are, we think, long-term going to be genomics and nutritional based. So one of our primary investment thesis is that we have a gap in nutritionally dense food. And what we've learned on the campus is that polyphenols, which are like the colors in fruits and vegetables, right? The orange in an orange, the red in an apple, the yellow in a squash. These are polyphenols, and we've learned on campus that these largely govern gene and gene expression. And so those are important findings. And on this campus, we have UNC Charlotte, Duke University, NC State, North Carolina A&T, UNC Charlotte. And by order of the state of North Carolina's legislature, if you were a public university, you located your genomics and nutritional departments here in Kannapolis, North Carolina, which is a suburb of Charlotte. So if you get a master's or PhD in nutrition from Chapel Hill, which has the #1 ranked nutritional research institute on the planet, you don't go to Chapel Hill to get it. You get it here in Kannapolis. And they have a phenomenal group of researchers, principal investigators here on campus that are doing really, really amazing work at the intersection of human health and nutrition and genomics. And so those are sectors that we're highly interested in.
Yeah, and it's fascinating stuff. I think it's so early on in the research in those areas, but there's so much potential. I think in 20, 30 years time, we'll look back on the work you're doing and the people around you are doing as groundbreaking and change the way we look at healthcare altogether.
Well, we know that molecular diagnostics is coming as a major diagnostic tool. So right now you get blood draws and you get your basic panel of chemistries, lipids, hemoglobin A1Cs, potassium electrolytes, all that type of stuff. Well, where the world's going is typically with disease, you get some type of expression prior to a disease. So take lupus, for example. Lupus is an autoimmune disease. Well, we've just recently made an investment in a company that has the ability to see a lupus flare taking place 6 to 8 months before it actually happens, because they understand the underlying gene expression that's taking place that causes the disease. So in the case of many of the autoimmune diseases, whether it's rheumatoid arthritis or Crohn's disease or many of the syndromes that you hear of, irritable bowel syndrome, these are caused by the immune system being ramped up when it shouldn't. And so in the case of lupus, the immune system ramps and it starts eating away at your organs slowly. And over time, that deterioration, that digestion of the immune system of your organs, pricking at your organs, causes a flare. And in order to shut off that flare, you usually use powerful drugs like steroids to shut off the immune system to stop that flare. Well, if you can see it happening long before, then you don't have the $40,000 hospital visit, because by the time you get in a flare, it's very serious. And so we're seeing this ability to measure what's going on really early in disease and disease states with molecular diagnostics and testing of DNA. So one of the investments that we made, actually one of the acquisitions that we made, is a company called Aramid Genomic Services. And so we provide a variety of services to a number of clients, like the Gates Foundations, the NIH, Duke University, Chapel Hill, University of Nebraska, are clients. And the genomics CRO that we own does method development for a number of biotech companies and other initiatives and principal investigators. So we're actually engaged with working with principal investigators to help them understand how best to take their concepts and make it into a laboratory development test and ultimately a CLIA test that can be used by physicians to look at disease much earlier. So we're going to see a lot of improvement in the diagnostics of oncology. We're already seeing that with liquid biopsies. We're going to be seeing a revolution in the dementias like Parkinson's and Alzheimer's. We're working very closely with a couple of Alzheimer's companies. We're working very closely with a Parkinson's company. And these involve molecular diagnostics, where we're able to more rapidly and earlier detect that something's going on, and then use these discoveries that we're finding in the genes and what the genes are expressing that are causing these various diseases, how to shut off those expressions. And one of the things that we're highly interested in is the role of nutrition in impacting gene and gene expression. So one of the researchers that we follow is a gentleman by the name of Dr. Bruce Lipton. And Dr. Lipton was at Stanford in the 60s, was part of the team that was actively involved with understanding RNA, DNA, gene expression early on. He actually coined the term epigenetics. So epigenetics is a term. Epi is a word that means above, and the term that he coined was epigenetics, which means what turns the gene on and off.
Right.
And he actually discovered what turned the gene on and off. So what he did is he took a naive stem cell. So a stem cell is unique in that it can become any other cell in your body. It can become an eye cell, a hair cell, a skin cell, liver cell, whatever. So he took a single stem cell, cloned it, and 50,000 copies in 3 different petri dishes, and then manipulated the medium of the petri dish. And he grew bone tissue, heart tissue, and skin tissue. And he looked at his colleagues and said, "You guys think that the DNA or the genes control everything. And I tell you, it is not the gene at all that it controls what's going on. It's the environment that the gene is in." And that's an important concept, and that's part of our investment thesis, is you need to understand that genes don't express accidentally. They express because of the forces or the environment that they're in. So what's the environment that your genes are expressing in? The environment that your genes are expressing in is the quality of your blood.
Yeah.
And what's the quality of your blood? It's what you drink, what you breathe, what you eat, and what you think. So it's very important for us to have clean water, clean air, be careful what we're exposed to, be careful that we eat ripe fruits and vegetables, and be careful what we think, because what you think, the brain is the most complex pharmaceutical manufacturing plant in the world. And so if you take a kid that was in Afghanistan, shot at for a year, and bring them back after being at war for a year, and they're dripping catecholamines like epinephrine and norepinephrine because they're having PTSD and they're having nightmares at night, that shuts down gene expression. It shuts down growth hormone, and it shuts down the production of stem cells. The stem cell production are in your bone marrow, and that causes disease. So what you're thinking, what's going on in your brain, actually can cause physical disease.
Yep.
And so those are important concepts to know, and those are areas that we're highly interested in. And we think that the earliest indicators now are going to be the ability to precisely measure DNA and RNA expression. And so we think that we bring some value from an investment thesis because we're actively involved with that with an asset that we own at AirMed Genomics Lab, and we have a lot of principal investigators across the globe that we work with.
Yeah, it's incredible stuff, and I think like I said before, I think in a couple decades' time, because of a lot of this work, human beings may well be a kind of another level of consciousness, and physical health and mental health than we are now. Once people start to become more, and some people already are reasonably aware of this and so on, but once we start to become more aware of this and how we can really optimize our brains and optimize our bodies to be to be at a higher level of existence, I guess, than we are now. Maybe that will be the link to us starting to understand how to use our brains more fully, and everything opening up, and the next stage of evolution maybe even occurring. It's big stuff, but it's very possible. It comes at treating healthcare as from a completely different angle than I think a lot of Western medicine have done, which is like a reactionary approach when we get sick, rather than looking at preventative measures, and how we live in the environment we live in, as being like essentially very much the most important thing. But if you can get those pieces put together right, who knows? Who knows what's possible with the human body and human mind?
Well, we know that genes express very well to a good environment, and we understand the power of the genome. So precision guided medicine is here. There are a number of precision guided medicine companies that use the gene to determine what therapy is best, what approach is best. This drug will work if you have this DNA signature. This drug won't work if you have this DNA signature. So doctors are learning how to use precision data from the genome in order to best treat patients that are sick.
Yeah.
However, the idea of precision guided nutrition has been, we can say, a bit of snake oil. There have been a lot of claims around nutrition and genes and wellness and so forth.
Yeah.
But most people haven't tied that definitively to outcomes. However, we did make an investment in acquisition of some technology at the University of North Carolina in Chapel Hill, and started a company called S&P Therapeutics. It stands for Single Nucleotide Polymorphism. So nucleotides are the building blocks, the bases in DNA. There's 6 billion bases in your DNA, and they make up 25,000 genes totally. So you have 25,000 genes that can express, and those genes, when they express a complementary DNA that then makes a messenger RNA. That messenger RNA then is read by a polyribosome that then makes proteins.
Mm-hmm.
So you have your genes encoding for enzymes that then do metabolism. And so it's genomics, proteomics, and metabolomics, which are really the three omics that are a big deal that a lot of folks are looking at. Now, in the case of S&P Therapeutics, what we were able to demonstrate, or our researcher that we partnered with was able to demonstrate, was that there are certain enzymes that are needed to make certain nutritional items like vitamins or minerals and so forth effective in the body. So you take vitamin B9, for example, folate. Vitamin B9 is needed. However, folate is not active. It has to be metabolized.
Right.
And so there is an enzyme that adds a carbon onto the folate. Now it's called methylfolate. So a methyl group is a carbon with some hydrogens around it. And now it's active. Well, if you can't make that gene that adds that methyl group, that's a problem.
Yeah.
if you can't take choline, for example, and make acetylcholine or phosphatidylcholine, if you don't have the enzymes that do that, that's a problem. So what we were able to do is Dr. Steven Zeisel, the researcher that discovered these discoveries, was able to identify certain diseases that were caused by a patient's inability to make certain enzymes because they had mutations or mutants or variants in their DNA.
Right.
And so we definitively tied this back to 4 disease states using knockout models where we knocked the gene out in animal models, and indeed the very disease that Steve thought would take place, and then you feed the subject the correct nutrition and it corrects. So if you're having an issue with choline and you need phosphatidylcholine, or you're having an issue with folate and you need methylfolate, we can give you the methylfolate, and the issue or the disease that's in progress can be arrested. So it's really fascinating. So we're moving beyond precision-guided medicine into precision-guided nutrition, but based in the science that you have a disease that's caused by your inability to do what's called one-carbon metabolism or add the methyl groups.
Yeah.
And we're able to just like precision-guided medicine, give the right medicine to solve the problem. In this case, we can give the right nutrition to solve the problem, because indeed it's a nutritional issue, not an issue that's going to be solved by drugs per se.
It's incredible. Absolutely incredible. So I would, as I intimated at the start, like I would say not only some of the work that your existing portfolio companies are already doing, but the potential that you have to incubate, accelerate, add strategic value to, add like collaborative value to from a scientific standpoint, your collaboration with research facilities and universities around you is nothing short of extraordinary, John. I think like there's certainly room for more down there. I think if looking at investments into new portfolio companies, if they want to make the move over to Kannapolis, the value that you can add to people is extraordinary, without having to, build whole new labs, whole new facilities, whole new research relationships with different universities, wherever they may be. You've literally got it all right there. And that is so unique for a fund to have. Like, you're not only offering capital, you're offering so much more value add.
Yeah, so we are active managers in I think maybe I'm usurping you here with your thought, but maybe I can run for just a second on this thought. So we're active managers, and what we mean by that, Chris, is that we're not going to just make an investment and be passive. So we're investing in early stage companies. And we have a number of drivers. One of them is that we'll have the principal investigator participate in the company, but we won't have the principal investigator run the company. We want a commercial team running a commercial enterprise. The second thing that we do is once we do the seed investment, we require the commercial CEO to start soliciting strategic investors before we go out for our A round. And the reason that's important is we feel like we have a really good handle on novel mechanisms of action, and we feel like we've got a pretty good handle on whether or not there's a large commercial marketplace to support a commercial effort with this particular technology. However, the real validation of that is somebody that's already making a living in the sector that you're trying to move into. So you take, for example, S&P Therapeutics.
Yeah.
We seeded the company, and then we've had the good fortune of 6 major players in the sectors of nutrition and genomics co-invest with us. And one of them is a very large company called Balchem. Balchem's the largest producer of choline in the world, and a lot of the research at Dr. Zeisel focuses on choline and choline metabolism, and if they're the deficiencies in choline metabolism, what diseases is called. So clearly, there's an interest from Balchem in regards to their selling of choline.
Yeah.
And so that's really our approach. We're hopeful not to make bad investments, but we think that because we're early, there's certain risks that are involved.
Of course.
But we think that having a strategic come alongside us really validates that there is a commercial marketplace. And then the focus really is on, can you deliver the product and service? So in the case of S&P Therapeutics, for example, we spent 2 years doing the molecular diagnostic development of the test and got it scaled. And now we're in the preliminary scale of the sales and marketing effort with that particular molecular diagnostic test. We're super excited about it.
As well, you should be. Yeah, it's a great model, and as of course high risk, but you are genuine early stage investors, which is great to see. Because like you say, that valley of death, you call it, or
It's called the Valley of Death. That's
the
correct.
valley of death. It is a bloody great chasm sometimes. I'm try the amount of things that fall down before potentially amazing things can come to market is
But
amazing.
we have two assets right now um, that both principal researchers uh, tried to start a company,
Right.
and on both occasions failed. And one of the things that typically we have to explain to principal investigators is that the investment community, of which you and I are part of the investment community, we
Hmm.
represent the investor.
Hmm.
And while these principal investigators are brilliant, and in the case of both of these principal investigators that we're currently working with, one with some spiral steroids that we'll maybe talk about in a few minutes, and
Yeah.
with Dr. Steven Zeisel with the S&P Therapeutics, investors want commercial people prosecuting commercial plans. And so that's why we bring in a commercial team at the beginning. And that's very, very important
It is.
because investors expect that you understand not only how to deliver a product, but how to sell the product. This business is real simple. You have to be able to scale your product or service delivery, and you have to be able to scale the selling of that product and service delivery.
Yeah.
Getting to the scaling the delivery side usually isn't the issue. The issue is, can you sell it? And so I want experts that know how to sell products involved with the company early on.
Yeah.
And so that's one of our demands and requirements for us to come alongside a portfolio company.
Yeah, it is crucially important, and it's a mistake many make, and I do admire principal investigators, scientists, technical people who do try to take the step of starting a company and spinning something out and trying to make it work, trying to get it to market in a big way. But it is a very, very, very finite number of those people who also make good business people. It's a completely different ball game, and I think if
It's a
you've got
yeah,
you've got to bring in a chief executive to go alongside essentially a CTO, of which the principal
yeah,
investigator would become.
yeah. That's right. A CTO or CSO, Chief Scientific Officer, and
Yeah,
and that's typically the case. Now there are a few, and we know a few, and there's particularly one at Stanford that we like a whole lot, Dr. Jeffrey Glenn. He's had great success. He's in his
yeah,
fifth startup and has been there and done it. So there are exceptions to that rule. But typically, when
sure.
we find these early stage assets, it's most of these folks have not had commercial experience before. So we actually had the good fortune a few months ago to acquire some assets. I got a call from a principal investigator at Stanford, and he said, "Look, I've got a friend who's been in the lab for 40 years working on some novel stuff, and I think you're uniquely qualified to take a look at it." And so this is really interesting technology. So this particular investigator, when I called him, his name's Dr. Fred Casilo. Um, when I was talking to Fred, Fred said, "Look, I found 12 new steroids. 12 new hormones. And I'm like, you got to be kidding me, right? Okay. The reason that that's important is the Upjohn company, the first company I started with,
Mm-hmm.
learned how to scale the commercial manufacturing of steroids. And so I had the privilege of selling a lot of the steroids that you know today, or hormones that you know today:
Yeah.
testosterone,
Yeah.
estrogen, growth hormone, prednisones. Yeah, we sold Medrol dose packs, all that type of stuff. But there essentially 74 hormones or steroids that are kind of known in the endocrine system, and
Mm.
no significant discoveries in the last 50 years. So when this guy said, I found 12 new hormones, I'm like blown away, right? Because we know hormones are very powerful signalers in the body.
Mm-hmm.
And indeed, when I looked at what he was doing, these were so steroids are cholesterol based.
Right.
Okay, so the scaffolding of all steroids are cholesterol based. This particular these particular steroids had a choline attached to them, and because we'd already done an investment in a choline company, we knew a lot about choline,
Yeah.
and so it was really unique. So I asked Fred. He said, "Look, these are choline based steroids." I said, "Fred, do they work in pregnancy?" He said, "How the hell do you know that? Nobody should know that." Well, I'm working with a researcher that has been demonstrating that choline deficiency impacts the IQ of a baby. Okay, so that's Dr. Stevens Eisel demonstrated with Dr. Marie
Mm.
Coddle out of Cornell University. They measured the serum choline levels of women during pregnancy, and 7 years later measured the IQ of the baby, cognitive processing of the baby, and it was significant relative to the neural cognition of the baby, um, essentially equal to like 15 points on the IQ test with a
Right.
p-value of 0.02. So the levels of choline that the mother has during pregnancy impacts the neural architecture of the baby's brain. So
Mm.
when Fred said, "Hey, these have a choline attached," it was like, "Okay, are they working in pregnancy?" He said, "Absolutely." He said they govern preeclampsia. One of the steroids governs preeclampsia. One actually is important for um, NEC, which is a type of colitis that babies can get, and one is used in the ongoing neural tubal formation of the brains because it's excreted in the breast milk, and so it's needed. So we're currently in a development campaign to develop. There are three primary targets in those areas. We'd love to see a breast milk supplement, including gallatropin, because right now, Enfamil and none of the companies that are selling um, um, breast milk supplements
Mm.
are have gallatropin in them, and we know that's important for neural architecture of the baby. So we do believe that we can improve the long-term outlook for humanity, and particularly the IQ of humanity, by making certain that mothers get the appropriate nutrition and making certain that people are exposed to nutrition, particularly highly dense nutrition, because ultimately it's our conviction that these polyphenols and phytonutrients really govern gene expression. And when you have good gene expression, it's really hard to get sick. I would say that most illness can be corrected with appropriate nutrition. Not all disease, but many diseases can be corrected with nutrition. Matter
Yeah.
of fact, there's a really important book that Dean Ornish wrote many years ago where he demonstrated that by eating oatmeal you can actually reverse coronary artery disease. Now, when I read that book in 1992, I was blown away because I was selling a drug for uh, myocardial infarction, heart attacks,
Yeah.
and it was a clot blocker, like TPA, and it was like, if you have coronary artery disease, you're going to get a heart attack, and it can't be reversed. Well, he definitively showed that by eating oatmeal, you can reverse heart disease, and indeed, when you eat a good diet, you can actually reverse many of the illnesses that we have, um, including type 2 diabetes and coronary artery disease and many other diseases.
Yeah. I'm curious, John, because this is a very, I mean, in some ways, this is a very subjective topic, like that, what a good diet actually is. How would you define a good diet?
Well, I think I agree with you. I think there's that's up for debate, but
Mm.
I like to say, and this is my opinion, I like to say there's seven food groups. Okay, and the seven food groups are fruits, vegetables, legumes, beans, nuts, mushrooms, onions, and sunlight. Okay,
Let your
now
skin
let me
eat too.
yeah. So let me give you some anecdotes around this and some
Oh,
fascinating science around a number of these issues. Most people don't realize that mushrooms are unique and that uh, their metabolism isn't driven by carbon dioxide. We know that plants ingest carbon dioxide, but mushrooms ingest oxygen. And when you flip them over and you see the gills that they have underneath the mushroom, and so they're very unique, and they have a number of phytonutrients that are very, very powerful, including a number of anti-cancer nutrients that are important that help govern gene expression.
yeah.
The other thing about
I learned a few years ago actually through a documentary called Fantastic Fungi on, I think it was on Netflix. When I watched that, I learned on there that essentially mushrooms and human beings are so similar in how they're alive, and like literally breathing oxygen thing. I presume is like probably the biggest sign of that.
yeah, it's it's rather unique, right?
Yeah,
So we can ingest something that actually metabolizes oxygen instead of carbon dioxide.
yeah,
The other thing that's fascinating is when you look at, for example, COVID.
yeah.
Okay, so we know that COVID was an age-related disease,
Hmm.
right? And when you look at the technicalities of what does it mean to be age-related disease, so it's typically correlated with age, but more importantly, it's actually correlated with biological age. And
Mm.
we can now measure your biological age by measuring um, the caps on DNA, which are called telomeres. So telomeres um, shorten every division. And so there was a Dr. Hayflack out of Stanford that showed that a cell could divide about 60 times, and then the telomere, it's kind of like a reverse rattlesnake. As a rattlesnake gets older and sheds its skin, it adds, a rattle
Yeah.
every year. Well, telomeres are just the opposite. Every time a cell divides, the telomere gets shorter, and then ultimately you don't have any more telomere left, and your DNA can be damaged.
Right.
So we know that as people get older, that their DNA and the ability for DNA to repair gets impacted.
Mm.
Well, that greatly impacts one of the two important immune systems. So you have really two immune systems. You have your innate immune system and your adaptive immune system. So your
Hmm.
adaptive immune system is the immune system that treats flus and that type of thing, where you create your own antibody to go attack a particular virus. And that typically takes about 10 days to work. But your innate immune system is when you cut yourself and you see this white pus come in. That white pus are white blood cells, and those are a group of innate immune systems that immediately attack the virus or bacteria or fungi or whatever's going on.
Mm.
Well, in the case of children that have a very robust innate immune system, they typically don't get COVID.
Hmm.
And in young adults that had very robust innate immune systems, and in older people that were very healthy and had a very healthy innate immune system, they typically didn't get COVID.
Hmm.
Not impossible, but typically didn't. But the devastating effects from COVID came because the innate immune system wasn't working correctly, which is a natural defense against a common cold,
Right.
a natural defense against a COVID type virus. And so those understandings are important relative to understanding the importance of the seventh food group, which is sunlight.
Hmm.
Because when you get direct exposure to sunlight, your vitamin D levels go through the roof. which support your innate immune system.
Yeah.
Furthermore, there's also recent research, this is really interesting, that if you look directly into the sun in the morning, where the near infrared light spectrum is different, that you lower your cancer rates by tenfold. And the mechanism of action is absolutely fascinating. So it turns out that this infrared light spectrum turns on melatonin production in the mitochondria of the cell. Now, there's mitochondrial production in the brain that puts you to sleep. Melatonin production in the brain that puts you to sleep, but you need melatonin during the day, and that comes from your cells, and that's largely turned on by this near infrared light spectrum. So waking up in the morning and looking at the sun is good for you, and it's healthy for you. And so I could go on and on about the importance of having direct sunlight exposure. And there have been a number of studies in melanoma, in fair-skinned people, people of Scandinavian descent, people that you would think would get all sorts of cancers if they were exposed to sun. And it's actually just the opposite. All the literature shows that those that have melanoma that are exposed to sun actually have a better response rate. Those that got more direct sunlight during the day typically had a lower incidence of melanoma. So we consider sunlight one of the direct food groups. Now, frankly, if we really look at the energy in fruits and vegetables and nuts and mushrooms, that all ultimately comes from the sun. And the best fruit and vegetable for you is a fruit and vegetable that's been vine ripened. So if you look at a strawberry that's been vine ripened on the back of a truck where a chemical's been released, and you go pick that up at your grocery store, local food market, or you go up here to Kerrigan Farms, which is about 6 miles up here, where they let their strawberries ripen on the vine, and then you measure vitamin C levels and other phytonutrients and polyphenol levels, there is no comparison. The density of nutrition in a vine-ripened fruit is 10 to 40-fold higher. So it would be like taking a Motrin, but having a generic Motrin where you're trying to take 800 milligrams, but it only had 10 milligrams in it. The fruits and vegetables that we have today don't have the amount of medicine or polyphenols and phytonutrients that are needed to govern gene expression. So we're highly interested in highly dense food, or supplements that can replace that can help govern gene expression. We think those are important findings. We're investing in those type of initiatives and the commercialization of those initiatives, and being on the North Carolina Research Campus gives us really unique insight into a number of these initiatives. Now, the National Institutes of Health last year announced that they were making a $1.8 billion investment in precision-guided nutrition. And that's being led by Dr. Susan Sumner on our campus that we have a great relationship with. She's at the Nutritional Research Institute at Chapel Hill, which we have already acquired technology from. So we're very close to this sector. So when I say that we're active investors because we're working very closely with the people that are doing the research. We just don't read the research. We know the principal investigators, and we're interacting with them at a number of very prestigious universities in the areas that we're highly interested in helping humans understand more about. And ultimately, the commercialization of these are going to increase the knowledge and the importance of appropriate nutrition, the importance of vine-ripened fruits, and while there's human health that can take place so we don't have to have disease care. Now, Western medicine is very good at sick care, but Western medicine is not very good at keeping people well.
Right.
And we have all sorts of problems in our not only in our food channel, our food distribution networks, our food chain per se, and it's not malicious. I just don't think fundamentally people understand what food is, and so we have an issue. And until we describe it in a language that people understand, which is our language is a language of science, this campus is really codifying why fruits and vegetables are good for you. And we're interested in making those discoveries commercially available to people to help them live healthier lives.
I'm sure it'll be very welcome. I think the work you're doing is incredible, John. I'm still blown away by what I saw and what I experienced visiting with you. It is truly a pleasure to be working with you and supporting you and the funds and portfolio companies. And I really look forward to working with you for many years to come. Yeah, I think we could talk all day, but you probably have many things you want to be getting on with. So I think we can wrap it up there. Although I would like to definitely have you back on again at some point in the coming months to talk in more detail, and hopefully talk about some new portfolio companies being added as well, and the work continuing. So until then, it's been a pleasure having you on, and I'll bid you farewell. And as I say, very much look forward to continuing working with you. And seriously, thank you from the bottom of my heart, and I think it should be from the bottom of everyone's heart for the work you've done in your life and the work you continue to do. You're an incredible person, and thank you very much for being here and doing this.
Well, thank you for your kind words and thanks for hosting us and letting us get our story out there. We were delighted to have you on campus and for you to experience firsthand what we've built here and how we think that our approach at Coddle Creek Capital and the active management really helps create alpha for our investors. So we have the privilege of working with wonderful principal investigators. We've got a wonderful campus, and the more that we can expose it to people and investors about what we're going on, the better from our standpoint. So for that, we're grateful, and thank you for hosting us and helping us get our message out there. Thank you, Chris.
My pleasure, John. Thank you very much.
Cheerio.
Bye, everybody.